So far, what we've done here is actually batch and inoculate a mini. So now, later in the process, when you want to start to learn about what's going on inside, this is what you would do to actually pull your samples to do any sort of offline analysis that would be required. So there are several reasons why you may or may not need to take samples. Some of them are cell counts, imaging, cryopreservation samples. But in any case, the important part is that you take a representative sample. So we're going to talk a little bit more about that in detail here. One of the most important things to consider when you're taking samples is the volume and the frequency of the samples. So in our 0.5, we have a lot of space to work with. You can take a lot of samples over the duration of your culture. But you have to remember, when you're working with something smaller like our PBS 0.1, you can't take as many samples, but you can do a lot more minis. So now we're ready to take a sample. I've moved my mini vessel into my BSC here, but in order to take a representative sample, I need to start mixing it. And to do that, we're obviously going to put it on our PBS Mini base. It's recommended that you have a dedicated base inside of your BSC. That way you don't have to be cleaning and transferring every time you want to take a sample from your incubator to the actual BSC. I'm going to place it on its dedicated mini base here. And you can see the vertical wheel actually kind of rotated and locked into place. So that's just the wheel coupling to the magnetic drive system of the base itself. So I'm going to increase the speed here to match our culture speed. And then we're going to talk a little bit more about why the RPM is so critical for sampling. All right, so here we have a bit of an exaggeration shot for you. So you can see with a lower RPM, we have what we call dead zones here. So you can see even with this lowered RPM, we still have really good mixture down here in the normal culture area. And this would actually be fine for culture as long as there's nothing settled along the bottom. But for sampling, since we want to get a representative sample, we want to make sure we don't have any dead areas up here. So what I'm going to do is I'm actually just going to bump up that agitation. So I'm just going to bump it up a few RPM at a time so I can make sure to constantly check. And so you really only have to do this the first time you're working with your culture set, so that way you know how much to increase it by the next time. So we can see here, I've bumped it up to about 20 RPM. And you can see they're still present, but a little more closed. So I'm going to bump it up a little bit more. And since this is only for sampling, it's a pretty short duration of time that we're doing this increased RPM. So it's not going to have a huge impact on your culture overall, but it's really important that we do this to get the representative samples that we need for our data collection. So here with 30 RPM, we can see that we're nice and tight, pretty closed all throughout. So now we're going to be able to sample using 30 RPM for this particular setup. We have our mini vessel now agitating at our increased speed, so that way we can get rid of these dead zones up here. So I'm just going to pull a sample. And the two most important things about taking a sample are going to be the position. So we're going to put the tip of the serological pipette right around this crest here. So we're not going to hit the wheel, but we don't want to go too high up. And the second most important part is going to be taking your sample as a single pull. And that's all because we want to be representative here. So I'm going to go in there, keep this nice and straight up, and in this case, I'm going to take a 5 ml sample. So I just want to get as close to 5 as possible. So in this case, I'm off by 0.2, and I'm going to leave it as such because I can always do the math afterwards to figure that out. So I have 4.8 exactly. We're going to dispense this. And there you go. You have a sample to do anything that you need.